Good afternoon. I am Sriram Subramaniam, an investigator in the Laboratory of Cell Biology at NCI, and I'm delighted to be introducing our speaker today, Dr. Eva Nogales. She's currently an HHMI investigator and professor in biochemistry and molecular biology at the University of California at Berkeley. Eva received her BS in physics and solid-state physics in Madrid, and then did her PhD at the University of Kiel in the United Kingdom, and then eventually moved to Berkeley to be a postdoctoral fellow with Ken Downing, and then she rose up the ranks at UC Berkeley and now is professor there, as I said. Eva's laboratory uses cryo-electron microscopy to visualize structure and dynamics of large protein complexes, with a particular emphasis on looking at those in the cytoskeleton and also the machinery involved in gene regulation. She's been doing some very exciting work in defining how these dynamic molecular machines that are central to life work, and I think you will see that she's as dynamic a speaker as some of the assemblies that she will talk about. Anyway, please join me in welcoming our speaker for today's lecture. The title of her talk is Visualizing the Molecular Machinery Controlling Human Gene Expression. Welcome, Eva.
Okay, thank you very much. It's a great honor to be here. It's actually a greater honor that I thought until the point where I went to Jackie's office and she showed me this plaque with my name, and I thought, oh my God, this is a big deal. So I hope I stand up to the occasion. So today I want to tell you a number of vignettes on our studies of human complexes that are important for regulating gene expression. And as Shiran said, my lab works in two completely different systems. We work on cytoskeletal components that are important for cell division. We've been studying microtubules for a long time, and also more recently septins, so self-assembly systems that are involved in mitosis and cytokinesis. But the other half of the lab works on machinery that are involved in the central dogma. And the only connection between the two areas of the lab is that we use, as a methodology for getting structural and dynamic information of these systems, cryo-electron microscopy and image reconstruction, which is ideally suited to study the complexity in size and dynamics of these two completely different systems. So today, I'm going to be concentrating on work that I hope is recent, is either recently published or unpublished work, although in some cases we've been working on this for a long time. So this is a little bit of the overview of what I'm going to be covering. I'm going to tell you about, fortunately, this is hardly visible, or is run out altogether. Hold on. Let me see. Can you guys see that, or do I have to use a different laser?
Cannot see it.
Not see it? All right. What about this one? Okay, I have to use two hands. I'm going to be in trouble. All right. So I'm going to start by telling you about the Polycomb Repressive Complex 2 and what we've been able to do for this essential silencing complex in terms of defining its architecture and what that has told us about how PRC2 functions and is regulated. And then I'm going to move on to our efforts to describing the very complex machinery that is involved in the critical step of transcription initiation at gene promoters. And I'm going to tell you first about TF2D, which is the component that is involved in recognizing key signatures at the start site of the gene. And then I'm going to tell you about how from that recognition step, the rest of the pre-initiation, transcriptional pre-initiation complex is assembled. So very quick biological introduction to the Polycomb Repressive Complex is that it's involved in silencing genes by tri-methylating lysine 27 on histone H3. Through this methylation, the PRC2 complex brings into chromatin a second polycomb complex, PRC1, which further modifies histone tails by ubiquitination. Now, these complexes of these modifications define epigenetic regions in the genome that cannot be transcribed, of heavily compacted chromatin. So the work that I'm going to be telling you about was carried out by a postdoc in the lab at the time, Claudio Ciferri, who is now a scientist at Novartis in Boston. And although the complex comes in slightly different flavors, he set up for the most abundant and probably critical silencing version of this complex containing 4 protein, ECH2, which contain the enzymatic activity, which is present in this set methyltransferase domain. This protein is very large, 751 amino acids, and contains other defined domains, including these 2 subdomains that are supposed to be important for the activity of the set domain. Additionally, the other large protein within the complex, SUS12, contains another recognized globular folded domain, the VEF's domain, which is involved in recognizing histone modifications that mark sites of transcriptional activity, and via interaction with these histones in cis, this VEF's domain engaged with these histone tails will repress the activity of the set domain. So this is going to be important for setting the limits of the epigenetic marking that PRC2 is able to confer. On the other hand, the protein EED, which is a WD40 domain, so it forms a beta propeller structure, is known to bind precisely to the histone marker generated by the methyltransferase, to the tri-methylated lysine 27 on H3, and this results in the transactivation, the activation of the set domain in trans for another histone tail, and that's what will result in the spreading of this transcriptional repression marker. And additionally, there's a 4 component that is also a WD40 domain that binds unmodified histone tails and just gives the complex general affinity for chromatin. So when Claudio put together this complex by expression in insect cells, he found that it was biochemically very well behaved, but when we looked at it in the microscope, it turns out to be so dynamic, so flexible, that it was really very difficult to arrive at a structure. So he looked into the literature for coactivators that enhance the activity of the complex, thinking that maybe via interaction with these cofactors, the complex may become stabilized. So he chose ABP2, and this protein did make a difference. The complex became more stable. Biochemically, the complex is very pure. It's about 250 kilodaltons and a 1-to-1 combination of these factors. So he looked at it using negative stain electron microscopy, and he did this rather than cryo-EM. Just let me tell you briefly, because the size of the complex is a little bit limiting in terms of signal for cryo-EM studies. So his negative stain images were utilized to generate class averages that had good signal to noise and showed us that now the complex seemed to be in a fairly defined conformation. It's just a readout of the sharpness of these class averages. And then using 3D reconstruction methodology, arrived at the structure at about 20 Angstrom resolution of this complex. And this was has been the only structure that we yet have of the complex as a whole. So I told you that 2 of the components of the complex are WD40 motifs, and those at this resolution will look like donuts, and this is exactly what we see for these 2 areas marked as A and D and colored in blue. But which one of them is EED versus the other protein is impossible to tell. So this is a situation in which the resolution is limited. We get a shape for the complex, but we haven't yet learned anything about how the complex is organized and how these factors come together. This is a situation in which EM scientists find themselves quite often, especially at the beginning of a project when the resolution is limited. So what do we do to extract biological information out of low to medium resolution structures, so say between 8 and 30 amino acids in resolution? So one of the things that we would like to do, and is always incredibly informative, is to dock into the structure available crystallographic models of some of the components. In some cases, this is fairly trivial and easy. In others, we need a hint of where those subunits are to begin with to be able to place these pieces of the puzzle into the density. So localization of subunit by labeling will be a good thing to do, both if you have crystal structures or if you don't have them, just to basically map up the organization of the complex at this resolution. So one possibility that people have used in the past, including ourselves, is to use subunit-specific antibodies. And these work from time to time, but in generally the binding is non-stoichiometric. The affinity of these antibodies, even those that are used for affinity purification, for example, is not ideal. Ideally, what you would want is a one-to-one labeling that can be obtained by genetic tags of the type that we use all the time in molecular and cell biology, such as MVP, GST, or GFP, to mention some. So there's an obvious process for using these kind of labels, where the idea is that the mass of this protein will appear as an addition with respect to the wild type, and the position of the tag will therefore indicate the position of the termini of the protein that you have labeled. So the pluses are that these kind of tags are readily available. They're part of our molecular biology tool set. Also, they have a size of the order of 40 kilodaltons that makes them readily visible by EM. So they're not too small. But on the other hand, they're also not too large, because you can imagine if I have a 250 kilodalton complex and I want to pinpoint where the position of the domain of a protein within the complex is, I don't want something that I can see very readily, but is the size of the whole complex. So this is about, these tags are typically at the right size. They're typically between 2 and 20% of the size of the complexes that we investigate. There are some minuses that you can imagine, and one is we are tagging a complex for which we don't know the structure a priori, so we may be placing a tag at a place that is important for protein-protein interactions, and the positioning of the tag may actually interfere with the formation of the complex. So one of the things that we can do, and this is what is done when these tags are used for purification, solubilization, or localization purposes, is we can put some stretch of amino acids, a linker region between the protein and the label. But that will, in many cases, facilitate the reconstitution of the complex, but it ends up resulting in the tag being too flexibly attached. You can imagine that if that linker is flexible, the movement of the tag now will not allow you to localize its position very accurately. So there is a compromise. You have to play with the two in order to find an optimal situation. Let me tell you, although I'm not going to show you examples today, that we can also use a stable deletion mutants. That will be producing a complex that is missing one of the subunits at a time, and if removing that subunit doesn't interfere with the formation of the rest of the complex, in that case you will visualize the absence of the density, which again will pinpoint to where its position is in the wild-type complex. So let me show you this little bit of experimental results. These are class averages of the PRC2 complex, and I'm showing this little movie where we are superimposing class averages that correspond to the same view of the complex, but I want to show it to you for two reasons. One is that I hope that you can see this region right here. That's an MVP tag that is moving around, but very clearly is connected to the complex through this point. So that's exactly what we are labeling. This is the position that the MVP is marking. But I also wanted to show you this because it gives you an idea of the flexibility that exists in this complex, even in the presence of the stabilizing ABP2. And it may be the reason why our resolution is still limited. Okay, so what Claudio went about doing very systematically was to place either MBPs and N and C terminal ends of each of the proteins, or something that had never been done before for EM labeling, which is to place GFPs internally, because GFP is N and C terminals very close to each other. You can actually place it within loop regions in the structure between folded secondary structure elements, and see with more precision where certain domains, certain regions of the protein are. So this is dissecting beyond just marking where the N and the C terminus is. I want to show you this example to start with because it was like a proof of concept. These are two GFPs that were placed within the EED protein, which is one of these WD40 proteins, for which actually the crystal structure exists, okay? And there were two of them that worked. He placed it all over the place, but some of them did not give rise to an unstable complex. So these are the two that work between the first and the second blade, and between the second to last blade. Additionally, the crystal structure was solved with this protein bound to the N terminus of the enzymatic component, the ECH2. So by putting an MBP at the N terminus of the ECH2, we had another label, a third additional label marking the position of this subunit, okay? And this is what he obtained. So as you can see, by the way, so this is an example of the same view of the wild type. This is now the complex with the extra density of the MBP that positions the density with respect to the structure. And he did this a number of times, and this is now what he gets. So we have, this is the crystal structure. These are the positions of our two labels based on where they were in the sequence. And this is the position of the MBP. So you have one label here, one there, and one there. This has localized this donut density as the one corresponding to EED, but now the position of these three labels allows us not only to identify this region, but actually to rotationally position it. So using information that goes beyond the resolution that we have. Because again, this GSP here is here, this one is farther up, and this one has come around, 1, 2, 3, around the structure. Okay? So this kind of a strategy was used for all of the subunits. I'm going to show you this, which is the ECH2, so the core of the complex, the enzymatic protein, where he was able to use 5 different labels, 1 N-terminal, 1 C-terminal, and 3 internal, to basically localize the 2 SUN domains at the set domain and come out with an actual path for the whole polypeptide chains as it meanders on the top half of the structure. So again, this was done for the other components, including the ABP2 that had stabilized the complex. And interestingly, ABP2 was localized with these 2 termini here and here, basically in the neck that connects the top and the bottom half, which are moving with respect to each other in its absence, but that become more stabilized by this kind of a stapling of the ABP2 in the complex. So let me tell you that we combine all of these EM studies with cross-linking and mass spec that gave us information on the relative position of lysines throughout the complex. And these 2 orthogonal, completely orthogonal type of experiments gave us information that was both confirmatory of each other, but also that complemented one another and gave us a very good idea of how the whole complex is now organized. So what we end up with is a definition, a positioning of every one of the elements, and we can take the 2 crystal structures that exist for the WD40 domains, as well as homology domains for the set and SUN domains to explain a significant part of the complex. And for the rest, we know the identity, but there's no crystal structures. So what have we learned? Does this structure make sense? And the answer is that yes, it does, because we can now position the two domains that act in a regulatory fashion on the set domain to explain how they repress or activate for the cis and trans tails of histone. So this is the Webb's domain that is placed very close to the set domain, where histone tail will be tri-methylated. If this histone tail contains positive markers for transcription, the Webb's domain will inhibit the activity of the set domain in this tail. On the other hand, this is the ED that interacts with the histone tail that has just previously been modified, and when it recognizes it, it might again allosterically activate the set domain for the trans, for a next door tail that lacks this modification but will receive it. Okay. So this is a little bit more of a speculation, but I like to show it because it helps us think and design new experiments. We know that the activity of this complex is stronger on dinucleosomes than in mononucleosomes. We know that ABP2 likes to bind to DNA, interchromosomal DNA, and to HP1, which will be also localized between nucleosomes. And we can come out with a model that may not be correct, but at least makes sense and incorporates this kind of information, where the two nucleosomes could be localized on opposite ends of the complex, one interacting with the set and Webb's region, and the other one with the EED, placing the internucleosomal DNA by the ABP2. So this is the kind of model that now we would like to test by directly visualizing the interaction of the complex with dinucleosomes. So this was the first example that I wanted to show you, but I'm going to move now to our transcription initiation studies. So this is a very, very simplistic cartoon that is actually not even correct in the few details that it's showing, but I'm going to correct it by showing you the real structure in a minute. So just let me explain what it means to define, which is the complexity of the transcription initiation process. And let me put this in the context of the fact that I have really simplified the problem, because I've eliminated everything that has to do, for example, with the chromatin context. So what is cut here that I eliminated is something that said nucleosomes, but I'm disregarding all of this. I'm also going to even disregard the fact that the formation of the pre-initiation transcription complex is activated via proteins that bind to gene-specific regulatory regions. And I'm just going to concentrate on the basal machinery as it recognizes start site sequences, core promoter sequences, and ultimately assemble a set of proteins, including the polymerase itself, and TF2H, which has a number of enzymatic activity, including the one that will allow the opening of the core promoter for loading of the start site into the active site of the polymerase. So this kind of a structure that includes over 50 polypeptides is what we are aiming for ultimately. But I'm going to tell you today are two stories where we split this difficult problem into two. One concerning the recognition of the core promoter sequences by the large TF2D complex. The other one concerning the recruitment of all of these factors and the opening of the core promoter, but in the context of a simplified version of TF2D that contains just the tata binding protein. So the first story concerning TF2D core promoter recognition was the work of Mike Cianfrocco, a graduate student in the biophysics program at UC Berkeley that is now a postdoc at Harvard. And it's a collaboration with Jin Kadonaga, whose lab generated a super core promoter that contains all known most of the known core promoter sequences into a single core promoter called the super core promoter that has has increased the affinity of TF2D for the promoter and also results in transcriptional activation. So let me start by telling you that Mike struggled with this project for a long time. We had previously obtained a structure of human TF2D. We knew that TF2D was flexible, and we had postulated that it undergoes some kind of breeding motion. But we never anticipated the degree of the flexibility that really exists. So Mike, for a long time, could see TF2D bound to DNA, but when he tried to obtain a three-dimensional reconstruction using the APO complex as a model, he could never get there. And eventually, after a lot of investigation and reanalysis of his data, he discovered the following. These are sequences of class averages of TF2D. The scale here is very different from what I showed you before. TF2D is a 1 million Dalton complex that contains over a dozen different protein subunits. And in here, there is one domain, which I'm going to be referring to as domain A. That's how we call it for historical regions, because it binds TF2A. This domain, which is 300 kilodaltons, about 300 kilodaltons in size, moves across the complex by more than 100 angstrom. Okay. So this is shown in a little cartoon here, frame by frame, for these class averages. This is the overall architecture of the TF2D that we have previously described. I will refer to this as the canonical conformation, in which we have domains A, B, and C, C being central, and standing for central, because in this structure is in the middle of it. But this domain, lobe A, can move all the way across until it's actually not contacted C any longer, but move across this central channel to contact B. This kind of observation can be quantitated. Mike was a biophysics graduate student, so he liked quantitation. So what he devised is just a scheme to represent with a single value, with a single number, the position of A with respect to the BC core. And when he went through his data set and presented this in a histogram form, what he showed is that that distance of A along the main axis of BC changed quite significantly in a distribution that was bilobal. And when he looked at this peak, they corresponded to, again, the canonical state and the rearranged state. Okay? So the complex is alternating between two major steps, each one of which actually has a certain breadth. So each one of which is flexible on itself, but two well-defined states. So what was interesting is that depending on whether these are the two conformational states that coexist for APO TF2D. And the thing that was fascinating is that if you use different cofactors, or if you use cofactors and DNA, you can switch and you can change the weight of these two populations. Okay? So most importantly, for TF2D, that is bound to TF2A, which facilitate binding to DNA, and the supercore promoter from Kandanaga, it was very obvious that there has been a shift from the canonical to the rearranged state. And as I'm going to show you in a second, it is this rearranged state and only this rearranged state that actually binds DNA. So the TF2D state that we had not previously described. Okay. So what I'm showing you here are structures of both the canonical state and in a second to the rearranged state that were obtained from a sample that contained just TF2D, so APO TF2D, and a sample that contained TF2D plus 2A plus the supercore promoter. Okay? And these two states look very similar, and the A lobe is bound to the C lobe. This is what is seen for rearranged states. So we have TF2D alone, so these two structures will be coexistent in a solution that just contains TF2D, is now in the rearranged state, where the lobe A has moved from here to there. But now, in the sample that contains both TF2A and supercore promoter bound to TF2D, he sees this structure. This structure looks, is like the rearranged state, but now it has extra density that corresponds to DNA. Okay? So, the DNA is binding to this rearranged state. So this is one case, again, where we are limited in resolution, but in this case, one of the main questions that we could ask and actually find answer for is how is the TF2D organized within the complex? Okay? What is the upstream and the downstream end? What are the different core promoter elements situated with respect to the protein complex? So to do that, this is just a schematic of the supercore promoter. This is the upstream end with the Tata box, INR, and the MTE and DPE core elements, and that's the downstream end. So what Mike did was to use a longer DNA construct that he extended on the upstream end, so next to the Tata box, do another reconstruction and see where the extra density for this extra piece of DNA went. And I hope that you can see, it's particularly obvious in this view, that extra density comes out of this side. Okay? So this would indicate that this is the upstream end and this is the downstream end. This will mean that the TBP that binds the Tata box will be in this lobe, as well as TF2A, which interacts with TBP. So to test that, he, in a completely different way, he used gold labeling. He first gold labeled the DNA in 2 experiments, in each case at one end of the DNA, and then he went through the process of analyzing the data and positioning the gold with respect to the structure. And he did this, as I said, both at the downstream and upstream end, one that will be on the side of the DPE MTE and one that will be on the side of the Tata, and he found them at opposite ends, one at this end and the other one just overlapping with the density for the A lobe. He also went a little farther and used gold label to label TF2A, again, because we're producing smaller TF2A recombinantly rather than TF2D, which we immunopurify from killer cells. We have some means of modifying it. So by labeling TF2A with gold, he could see that TF2A is, again, in this lobe and actually moves from the with the lobe A from the canonical to the rearranged state of the complex. So all of this now gave us an idea of where the DNA begins and DNA ends, and therefore we can place the position of these core elements within the context of the complex. And this was complemented by footprinting information from the Katonaga lab that very nicely showed the regions of protection by protein contacts and the regions that were accessible. And I just want to emphasize one point, which is that the INR, which contains the transcription site, and therefore is the region that has to be loaded into the active site of the polymerase, is right at the site where the DNA is being held between the 2 lobes on TBP. And we think that this is going to be important for in transcription initiation. So Mike was able to come out with this kind of dynamic scheme of how the conformational switch occurs and how it is affected by elements like TF2A or binding to the supercore promoter. But with emphasis from additional data that I'm not going to show you, it is very obvious that it's only this reorganized state that is able to bind to the DNA. And we have tried with different mutants of the different sites so that we don't have a supercore promoter, but effectively different types of core promoters with distinct core promoter elements, and they always bind the DNA in this reorganized state and going from here to here across the central cavity of TFTD. All right. So what we would like to pursue and what is now our working hypothesis is that activators and cofactors will act by regulating this conformational switch between a non-core competent complex for the binding to the core promoter to one that is. So different regulators can bind to TF2D and have different effects in different core promoters by being able to switch, regulate, fine-tune the switching between these conformations. Okay, so this is just a summary that shows you the kind of movement that the A lobe undergoes within TF2D, how the rearranged conformation binds DNA, how through experiments, like I told you, like the extension, we can actually position the ends of the DNA and ultimately localize the position of the different core promoters within the context of this DNA, TF2D engaged DNA. All right, and I'm gonna, the last vignette that I want to tell you is going back to this transcription pre-initiation complex. And I want to show you the work now on how this transcription pre-initiation complex assembles and how it loads the DNA into the active site of the polymerase. And this is work that is accepted, but it's actually not published, so it will be hopefully new for most of you. So this is work that was carried out by Yuan He, a postdoc in the lab. And what he did to pursue this was to both simplify the process by what I told you, substituting TF2D by just the TATA binding protein, the TBP that recognizing TATA elements in core promoters, and also by devising a beautiful system for purifying the different stages of the pre-initiation complex in an effective manner. And let me tell you that here the difficulty is that both the RNase polymerase and TF2H are very large complexes, each one of them about 500,000 Dalton in size, that cannot be reconstituted and that have to be purified from HeLa cells. Okay? So we have to make this as effective as possible, even at the very low concentrations of proteins that we have. So what Yuan devised was the use of a core promoter that was biotinylated downstream, so that he could bind it to a streptavidin-coated magnetic beads, then use the different factors, add it in a sequential fashion and up to whatever point he was interested in, integrate excess to the DNA, wash the beads, and then use a cleavage site to release the preformed complex on the DNA. Okay? And to tell you how things progress, he initially did all of these in negative stain reconstruction to test that this method would work. This is fast enough, but the resolution is somehow limited, and importantly, the DNA is invisible in negative stain. And then when he showed that it works, and I'm not going to show you that state, he moved into cryo-EM reconstructions, where the resolution of the structures that I'm going to show you are typically between 11 and 40 in Angstrom. What I showed you up to now was between 20 and 30, so this is an improvement. And this, in fact, was a resolution that allowed us to dock all the existing crystal structures with great accuracy and confidence, and basically gave us a full map of protein-proteins and protein-DNA interactions. So I'm going to show you a number of movies that show the process. Just let me tell you that the way he started was to use, again, TBP, which substitutes for the full TF2D, TF2A, which enhances the affinity, increases the affinity of TBP for the core promoter, TF2B, which has core promoter regions that it recognizes and that interacts with the polymerase, and polymerase itself. So this is a starting point. There was a great starting point because there are crystal structures for all of these elements, and the crystal structures comes in pairs. So there's structures of Pol2 with 2B, there are structures of 2B with TBP, of TBP with 2A, and TBP with the DNA. So in principle, our structure has to be able to be explained by all these crystal structures as they come together, with no excess density and with no clashes. Okay? And then after that, he added one at a time, TF2F, TF2E, and TF2H. That's what I'm going to show. Okay, so this is the promoter DNA. You can always see it. And the densities have been labeled with the color of the crystal structure that we could dock into the density. So this is the initial structure. And one thing that I want you to notice is that there's a large part of the DNA that is invisible. This is due to the fact that it is mobile, is not engaged with protein, and it gets averaged out. Okay? Otherwise, what happens with the crystal structures? So if we superimpose the crystal structures that exist two at a time, we come out with a model. The model almost is exactly to a density, but we have to make a little rigid body motion that is probably due to the fact that the crystal structures don't match exactly what happens in the context of the complex. But we have beautiful agreement between our structure and all of those. Okay? Now, when we add TF2F, we get extra density that we assign to protein. We have an extension of the density corresponding to the DNA that now explains the full core promoter that we use. And we have a number of movements that occurs in the existing pre-initiation complex. We can explain these two extra densities as the WH domain of RAP30 and of the dimerization domain within TF2F. So this WH domain is interacting with the DNA and with and basically completing a wrapping of the protein around this upstream DNA that looks very much like an inverse nucleosome, if you want, with the DNA in the middle and the protein around. And the polymerase itself, through the protrusion region, completes this protein-protein set of contacts that really keeps this downstream DNA very, very tightly bound in a very stable manner. And this is going to become very important in a minute. All right? So what I want to show you in a second is what are the changes that we see when we added TF2F. And I'm going to do that by basically, I'm going to show you the two structures in a second. Then I'm going to remove the densities and just concentrate on the crystal structures that have been docked into them. And I'm going to show you, starting from this structure, what happens upon addition of TF2F. So one obvious thing will be the additional mass of the TF2F itself. But there are a number of conformational changes that occur simultaneously. This region comes closer to the polymerase by the forming of this tight nucleoprotein complex. And the other is that the clamp domain has to come up, and that allows the docking of the DNA. Now, when we add TF2E, we don't see any further conformational changes. If anything, we see an stabilization of the complex that results in higher resolution. So there are regions in this density for the polymerase that are about 10 angstrom, so that we can actually start to see individual alpha helices. But interestingly, importantly, this resolution is enough for now to see the DNA a little bit better. So there's no crystal structure for TF2E, but there is a model of 3 WH domains bound to the clamp coils based of cross-linking data from Steve Hahn that we can take, superimpose with the clamp domain, and that qualitatively very nicely reproduces the extra density that we see, and that indicates that one of the WD40 domains interact with TF2E, and the other with elements in the polymerase that had been identified by cross-linking or identified genetic studies by their relevance to transcription and initiation, although it was not clear what it is that they were doing. So these are the two contact sites with the polymerase, and this is the contact site between TF2E and TF2F. The other thing that we can determine are now the positions of contact between the polymerase and this engaged DNA in the closed complex, both regions in the clamp head and the RPB subunit. And these contacts occur sandwiching the INR and the start site almost symmetrically, which is right here. Again, this will become important in a second. Okay? So these contacts that form as the clamp domain comes up actually slightly deform the DNA. So I'm going to show you this by removing some of this density, showing you just the DNA. If you just were to extend it as B form, this is where you will see it. But instead, we see a bending of the DNA that is marked by this position of these two interactions, which, by the way, are the only positive charges that occur on the surface of the cleft of the polymerase. So it makes sense that they're the one interacting with the phosphate backbone of the DNA. Now, something that we don't see when we do this rigid body docking of the crystal structures are two regions in TF2B and TF2F that must be very flexible. If they were to be present, as in the crystal structure, they will basically clash against the DNA at a very important region, the region where the bubble of the DNA normally starts to open. So again, this will become relevant in a second. So in this structure, they are disordered and they're invisible. Okay? So what I want to tell you now, so this is the pre-initiation complex up to TF2F in the closest state. So before the DNA is open by TF2H, by the helicase protein within TF2H. So we wanted to get an idea of how this complex looks in the open state, after bubble has opened. And to do that, what we have done to start with is to generate a mimic of that state by using a clever template. And this is a template that is based on Patrick Kramer's template for obtaining the crystallographic structure of yeast pole 2 in a backtrack state. And what we have is a very small region of DNA-RNA hybrid. And we have positioned the start site with respect to all the upstream DNA that binds the initiation factors in right here at this junction. So what this template does, is it allows the engagement of the active sites right here, but having a very, very small RNA-DNA hybrid, which will not have, therefore, pushed out all the initiation factors. So it will be the equivalent of a very, very early stage in initiation where the polymerase have not cleared the promoter. So this, in principle, if the idea is that you can have an open complex and still have bound initiation factors, should give us a structure where the DNA is engaged at the active site, but all the transcription initiation sites are there. So all the transcription initiation factors are there. Okay, so this worked, and I'm going to show it to you. So this is the structure now of this mimic of the open state, where the DNA has very dramatically moved now into the active site, but where all of the transcription factors, initiation transcription factors, remain attached. And I'm going to concentrate on the changes that we see, which are all very telling. Okay, so I'm going to do that, but actually, again, do one of these comparisons. In this case, we have all the same components in terms of protein, but two different DNA templates, one in the open state, one in the closed state. And we can basically superimpose the structures and the transcription initiation sites and all the upstream DNA, and all the changes have happened here, where the DNA has now rotated in plane and moved in to place the initiation, the transcription start site by the polymerase. And the one major change that we see in the protein is that now the clamp domain that had moved up to engage the double-stranded DNA now comes down as the bubble is open and the DNA reaches the active site. And in the process of doing that, it does something very interesting, which is that it moves the rudder domain within the clamp head, now making contact with the 2B and 2F regions that before were disordered, and now generating a change of protein density that is placed right in the middle of the DNA bubble, we think precluding the rehybridization of the DNA before transcription is really on its way. All right. So ultimately, of course, this is not how things happen. What happens is that TF2H binds and through its activity opens the DNA. So we added TF2H, and this is a structure that so far we only have in negative stain. The cryo-EM will be coming soon, hopefully. So the DNA is invisible, but I hope you can see that it's very obvious that you can just place the DNA as it was in the structure before, in the closest state with the rest of the other factors. And TF2H is engaging it downstream on the DNA. So there's 2 helicases in TF2D, XPD, which is supposed to play only an scaffolding role, and this is the one that is establishing contact with TF2E and polymerase, and the other one, XPB, which is the one for which ATPase activity is required, okay? And that's the one whose crystal structure we can fit in right at these sites where the balls are regions that have been identified through cross-linking with XPB in studies by others. So this is what we think. This is our model, is that in this position, TF2H is positioning its ATPase activity right here. This positioning is determined by the full complex making contact with the rest of the PIC. And now this is in a position where its translocase activity in this direction while making this contact will result in the translocation, in the movement of DNA in this direction. As the helicase move along the path of the DNA, it will generate super twists that will be relaxed by unwinding. Where can the unwinding happen? As this is being pushed, this part, as I told you, is incredibly tight. It can not only not move, it can also not unwind. So the unwinding will happen right here. It will be facilitated most likely by the surface chemistry of the receiving polymerase. But we think that as this translocates, the DNA is pushed. One of the points of contact remains. I don't know if I went very fast through that, but as we look at the open complex of the 2 contact points with the polymerase, this one is broken. This is where the bubble will start to open. This one remains. So the DNA is sliding, maintaining this point of contact, moving into the site, and relaxing the supercoiling by opening a bubble. As that opens, the clamp comes down, and this change of interaction occurs that maintains the 2 strands open during this process. I have just time to show you something else, no, I showed you that already. So the structure of TF2H that I show you is the one that corresponds just to the core complex. Additionally, there is a region that is very flexible in TF2H, which can take the CAC domain, where the kinase activity that actually phosphorylates the C-terminal end of RPB1 is located. So this region was too flexible, and when we obtained this reconstruction in the context of the PIC, we lose this by movement. But we could go back to looking at just TF2H by itself, and I'm going to be comparing it with the TF2H structure that we can just mask out of the full PIC. And when we look at the holo, at the TF2H alone, and we separate into different conformations, so you can see that this extra density with respect to this core, and again, through this class averaging, we can sort out a little bit, but I think hopefully you can see that this is still a little fuzzy, and this is due to mobility. Then we can reconstruct those, the TF2H, and see that there is this extra density with respect to the core TF2H that we resolve clearly, that is more stable, and we resolve in the context of the PIC. But we can take this structure now, superimpose the core, and see it in the context of the whole PIC. And that, I'm going to show you now that extra density and how it is located. And actually, this region, although it's much lower resolution, again, because of the mobility, fit very well with these two, locating the 2 major components of the CAC. And what I want to show you is that the position of the CAC is facing the position where the crystal structures lose track of the chain as this unstructured C-terminal extension that is phosphorylated is located. So we think that, again, this whole huge complex is being used to position both the helicase activity and the kinase activity at the right place within the context of the PIC. So just a summary of what we have seen. we've been able to obtain a number of structures that allowed us to see how the complex builds up, how the DNA is stabilized, how the engagement of the DNA requires movements within the clamp of the polymerase, and how ultimately addition of TF2H will lead to a translocation of the DNA and an opening of the bubble at the right side with elements of the polymerase now as the clamp comes back down, establishing protein-protein contacts that actually stabilize the open bubble. This is our working model. Right, so I just want to end by thanking all people involved. This work was funded by NIH, by an R01 from NIGMS, and there were some technical aspects that Mike needed to sort conformational changes that had been funded through a more technical general grant from Human Frontiers in Science program. We had a number of collaborators. Claudio got help from Gate Lander on all the image processing, and we collaborated with Eversalt on all the cross-linking that I very briefly mentioned. As I told you, Mike collaborated with the Kadonaga lab, both obtaining the supercore promoter, but also through all the footprinting that, again, I mentioned only very briefly. And Yuan He got help from Jay in our lab, just like Mike, because she's the one that purifies these large compressors from HeLa cells. TF2H, at that time, we actually obtained from Dylan Tajes at Boulder, and some of the recombinant components like TF2B, we obtained from Carla Inoue, which is in Robert Tijan's lab. And I thank you very much for your attention, and hopefully you have some questions or comments. Thank you very much.
Thank you, Eva, for a very exciting lecture. I am sure there will be some questions.
Very interesting talk. If I understood correctly, the opening of the DNA is at the expenditure of ATP, I'm assuming. And once you are changing the DNA structure, is the surrounding medium, for example, the proteins, are changing their charges. I noticed that there was a zinc in the active site of, I believe, RNA. And if there is a conformational changes by zinc and also during the rearrangement, is there a charge differences? The density would shift.
So you're asking me a very sophisticated question that we can address only indirectly by using the crystal structures of the yeast polymerase or a homology model that we have generated based on another map that I haven't shown you of DNA engaged human polymerase at about 9 angstrom using molecular dynamics building on the yeast. The resolution of these structures are not such that we can see this directly. We can only infer. And I think there is a very important point that I think you bring in and I mentioned very, very quickly, which is that as XPB is pushing on the DNA relatively far away from where the bubble is opening, the pushing and the movement of the DNA has to be directed by the chemistry of the polymerase receiving cleft.
Right.
Okay. So this is very interesting because it is, again, is all electrostatic. It has to be because there's no sequence specificity that is determined by the polymerase. Very interestingly, if you look at all the crystal structures, there's very little that can be seen of any single-stranded DNA. You can see a little bit of the DNA-RNA hybrid on the transcribed strand, none of the non-transcribed strand. So there is not a single position of any of these strands that anybody has been able to visualize. But the electrostatics are very important, and I don't know how we're going to see, because we cannot visualize it, not just because of a limit in our resolution, because crystallography has the resolution, but again, there's not a unique positioning of this, and definitely we're not going to be able to visualize atomic details of the process of insertion. This is going to have to be getting the best possible structures at the highest resolution of the two ends, putting the chemistry there, and then doing some careful, good molecular dynamics with a system that is huge and movements that are humongous. So to answer your question in detail is going to be very difficult.
I appreciate that.
And I would imagine that this information that may concern, that could concern mutations, for example, going back to analysis, but it is so hard to get mutants in the regions that are doing these that are not completely deleterious. So I don't know how much information can come from just putting all of that mutant data into the complex. It will be indirect, but it could add.
True. Thank you.
All right.
We welcome other questions, but before that, maybe I should remind people that there will be a reception at the NIH library right after the question, so please stick around if you can.
That was really beautiful work. And supercoiled templates at many promoters, TF2H is optional.
Right.
So I wonder if you could speculate about the mechanism for threading the DNA into the active site if TF2H isn't there to push.
Right. So in vitro, you can override the requirement for TF2H and TF2E, whose role is bringing TF2H at the right place, but basically using supercoiled DNA that basically allows for the formation of the bubble by itself. So again, in a way, you're asking me a question very similar to what I got before, which is the details on the mechanism of how that transition occurs. We can only, at this stage in resolution, we can only infer, and when it comes to actual dynamics of the process, it's going to be very difficult. But it is true that if you have, we are proposing that what TF2H does is just generate the supercoil that otherwise you introduce into the system by some other means, getting rid of TF2H. That's why that model is attractive rather than a more classical helicase kind of activity, which cannot, I think it's very hard to imagine that happens right at the site where this has to be inserted. It's just too crowded. So the helicase have to add at a distance. This idea of the helicase acting as a translocate is not new. It had been proposed before. It's just that now we have the kind of information that puts it into a structural context. But the details of that insertion, I think, is gonna take more than one technique, maybe something that has more of a time resolution information to get into, and it is it's very clear that the polymerase itself, that cleft is, and the interaction of the proteins that I tell you are going to be very important for determining the opening and the opening at the right site. Okay? So TF2F and this interaction with the RADA and TF2B are going to be critical, because TF2F is one that you definitely cannot get rid of, and is one that is absolutely essential for determining exactly the position of that start site. And that is just, again, is given by where it is in the context of all of these other factors that are holding the upstream DNA very, very tightly, under very tight hold. There is obviously some things that we can tell, but some others that we cannot tell when we are at 12 Armstrong resolution. We can only infer and then try to move from there, improve the resolution, and get more than one snapshot. So one of the things that we would like to do is actually dissect how TF2H works. So all that I'm giving you is kind of indirect. What we want is to add TF2H, add ATP, have the right sequence in the DNA so that you can start transcribing. So that we see the opening of the DNA really in a more physiological context. But also, if we want to look at very, very early stages, maybe play with the amount of ATP, with non-hydrolyzable ATP, with different nucleotides. That's exactly what we're going next. I mean, this is like a very broad view and the development of a system that hopefully will take us farther. Yes?
In your reconstruction of with the scaffold, did you see any stabilization of the trigger loop that was shown in Kornberg's structure?
Again, this is a kind of resolution that is, borderline. We will be able to answer the question. So a disclaimer that unlike what we have done for PRC2 and TF2H, where we're truly limited by the flexibility of the complexes, here we're only limited by the amount of data. So Yuan was concentrated on getting all of these states, not in pursuing one particular state to higher resolution. So the idea of pursuing both this last up to TF2E, which is the most stable of the complexes, with the two different scaffolds, but instead of using 50,000 particles, use 500,000 particles. We know that we can get the structure of the polymerase down to 9 or 8 angstroms, and at that resolution, we will definitely be able to answer your question.
Okay. Are there any other questions? If not, Eva, thank you again for a very stimulating lecture. And we have a reception in the library, as I mentioned before.